All right, today we're diving into one of the coolest, most efficient little systems in all of biology. We're going inside a simple bacterium, E. coli, to see how it makes an essential building block, tryptophan, with a kind of precision that would make any human factory manager, well, totally jealous. It's this beautiful dance of molecules, all run by one little stretch of DNA, the tryptophan operon. So let's see how it pulls it off. So how does it do it? Well, the trick is to think of the bacterial cell as this tiny, hyper-efficient factory. Its only goal is to survive and make more of itself. And to do that, it absolutely cannot waste a single bit of energy. Now, this factory has an assembly line just for building tryptophan, but here's the key. It only turns that line on when it's absolutely necessary. And the secret to that incredible efficiency, it's all in the design, a brilliant piece of genetic engineering called the tryptophan operon. So here's our game plan. First, we're going to look at the blueprint for this genetic assembly line. Then we'll check out its main on-off switch. It's super simple. After that, we'll uncover a whole second layer of control, which is where things get really clever. We'll see how it fine-tunes everything, and finally, how these two systems work together for one single purpose: absolute total efficiency. Okay, first things first. Before we can understand how the factory is controlled, we gotta look at the floor plan. Just like any smart operation, the layout is everything. So let's check out the genetic blueprint. And that whole blueprint is called an operon. This whole concept was so revolutionary, it actually won its discoverers a Nobel Prize, and for a really good reason. It's an incredibly smart setup where all the genes you need for one job, in our case, making tryptophan, are all clustered together and controlled as one single unit. It's like having the instructions for the machines, the power switch, and the safety controls all on the same page. Super efficient. So this single blueprint has three really critical parts. First up, you've got the promoter. You can think of this as the big green start button for the whole assembly line. It's where the cell's machinery, this enzyme called RNA polymerase, grabs on to start reading. Right next to that is the operator, which is basically the main control switch. Its location is key because it can physically get in the way and block the machinery from starting. And then finally, you have the structural genes. These are the actual step-by-step instructions for building the 5 different enzyme machines needed to make our final product, tryptophan. Okay, we've got the layout. Now let's look at the first, and honestly, the most powerful control system in place. This is the factory's master power switch. It's a system called Repression, and it makes a very simple all-or-nothing call. Are we open for business, or are we closed? And of course, every good factory needs a manager, right? Someone to watch the inventory. In our little factory, that manager is a protein called the Tryp repressor. It's made from a totally separate gene, and its one and only job is to decide when it's time to shut the whole assembly line down. And this all boils down to one simple question: how much tryptophan is actually around? Let's look at the left side first. When tryptophan levels are low, the cell is starving for it. Our factory manager, the repressor protein, is just kind of floating around in an inactive shape. It can't grab onto that operator switch, so the track is clear. The RNA polymerase can get right to work, and the factory runs at full speed, cranking out tryptophan. But now, look at the right. When there's plenty of tryptophan, this story totally flips. The tryptophan molecules themselves act like a key. They bind to the repressor, change its shape, and activate it. Now, our activated manager can lock onto that operator switch, physically blocking the polymerase. The whole factory grinds to a halt. It's this perfect feedback loop where the product itself becomes the off switch. So clever. Now, for a long time, scientists thought, okay, that's it, case closed, a beautiful, simple on-off switch. But then, in the 1970s, a researcher named Charles Yanofsky and his team noticed something weird. They saw that even when the main power switch was on, the factory wasn't always running at 100% capacity. That suggested there had to be something else going on. Another, more subtle control system at play. And the big clue they found was in the genetic messages, the mRNA that the factory was producing. They weren't just finding one type of message, they were finding 2. There was the long one they expected, the one with all the instructions for the enzymes. But they also found this other, really short kind of aborted message. It was only about 140 nucleotides long. It was like the factory was starting the job and then just quitting almost immediately. The big question was, why? Well, that little mystery led to the discovery of an incredible process called attenuation. And if repression is the factory's main on-off power switch, you could think of attenuation as the assembly line's speed dial. It's not about being on or off, it's about fine-tuning the speed of production based on even tiny changes in tryptophan levels. Okay, this is where it gets really cool, so stick with me. Let's go back to our first scenario. Tryptophan is low. As that mRNA message is being made, a little machine called a ribosome hops on and starts reading it. But pretty early on, it hits a spot that requires tryptophan to keep going. And since there's not much around, the ribosome just stalls. It stops and waits. And that pause is everything. It gives the mRNA strand up ahead time to fold into a very specific shape. We call it an anti-terminator loop. And that loop is basically a big green light for the RNA polymerase way down the line, telling it, full speed ahead, keep on going. But what happens when tryptophan levels are high? Well, that ribosome doesn't stall at all. It's got all the tryptophan it needs, so it just zips right through that section. And because it moves so fast, it actually forces the mRNA to fold into a completely different shape. This one is called a terminator loop. And this new structure is basically a giant stop sign. It forms right behind the RNA polymerase and literally knocks it off the DNA track. Production stops dead. And guess what? That's our short little piece of mRNA. It's the physical evidence of attenuation slamming on the brakes. So, what we have here are two totally different control systems. One is a big, powerful on and off switch. The other is this incredibly sensitive speed dial. But here's the beautiful part. They aren't competing with each other. They're working in perfect harmony to make sure the factory isn't just on, but it's running at the exact right speed. So let's just recap real quick. You have repression, that's the main on-off control, the factory's master power switch. It makes the big decision. Are we open for business or are we closed for the day? Then you have attenuation. That's the fine-tuning mechanism, the assembly line's speed dial. It's constantly reacting to tiny changes inside the cell, making sure that assembly line never runs a second longer or a tiny bit faster than it absolutely has to. And when you boil it all down, both of these incredibly elegant systems serve one simple, almost ruthless purpose: maximum efficiency. See, in the microscopic world, every bit of energy counts. Wasting energy building something you already have is a fatal error. And the Tryptophan Operon is just a masterclass in how life has evolved to avoid that at all costs. And that just leaves you wondering, doesn't it? The Tryptophan Operon is just one amazing example of nature's genius, found in a single common bacterium. It really makes you think, what other elegant, hyper-efficient systems like this are still hiding in plain sight, just waiting for us to find them in the code of life? Thanks for joining me for this one.